I'm really excited to have Professor Oliver Stegle here to give a presentation. He's really a leader in statistical genomics and system genetics, pioneered a lot of statistical machine learning methods in genomics and biology, and he has a PhD in physics, and I know him from a long time ago, where he did some mixed models, or talked about mixed models like 13, 14 years ago or something like that. Lots of fun. And you've done a lot of things, and now you're going to talk about advancing the tool chain for human genetics using AI and single-cell genomics. Thanks a lot, Oliver.
So I sort of have, I thought what could be useful in this audience, and basically also want to start with a sort of general integration twist, but then I think taking a slightly different chance into Caroline's talk early today. So, obviously we're really motivated also by this, ability of spatial assays, multimodal assays, and particularly the sort of ability of multi-omics, where we're now measuring in cells and tissues, different omics modalities, everything from transcriptome, epigenome. We had also, I think, a very nice rounded introduction to these different modalities that are coming up in this area. I would like to just sort of, give a bit of a sort of basic primer into some of the reasoning, the sort of approach we took at this, which is, I think, is slightly complementary to what we heard in the first talk. I think what I just want to mention as a sort of a prelude, which I'm not going to talk more about, but I think is very important, and some of the discussion went in this direction. Obviously, before we do any of this integration, right, we need to mindfully represent and process these data. And what turned out in practice, often a bottleneck is, what are the data structures? How do we store these aligned matrices? Even if you have like 3 omics layers, in the end, what's going to happen is you have thousands or even millions of cells from different experiments. But you need to keep track which of these matrices belong together. What are the physical connections between rows and columns? For example, if you have multimodal assay, or where things are disconnected, where you have maybe single-cell RNA-seq, single-cell ATAC-seq, but not from the same physical cells. And very often you have even combinations of this. So storing and representing this data is sort of a prerequisite. And I just, those interested, there's a very nice consortium, SCVerse, where we've also been contributing, and many others, and Mia Josef and Fabian Theis, and I think it's a very nice community effort where maybe it would be great to see more folks also getting interested in and thinking about solving these foundational problems before you can actually get to the modeling, which often turn out to be a major bottleneck and dealing with the data. So I will now, following the talk, put this aside and really assume, that we are representing multimodal data as, different modalities here in rows. So this could be one view, transcriptome, epigenome, proteome, whatever. And I really have this sort of use view and omic here, this canonically and exchangeably. But what I think is important, these are the different features we're measuring in different modalities. And sort of for simplicity, just assume they are representable as matrices. There are obviously subtleties to this. Not everything is a matrix, but you can also think about even shaping images to some extent in these dimensions and representing them, and think about, how do we solve different integration questions once we have these types of matrices. The other point I want to make is that, also in the column space, in discrete samples, you have different groupings. For example, different individuals. You might draw many cells from one person, cells from another person. And so obviously also the columns have some sort of structure. And there are boundaries, right, that are in some way made to be sensed. You could concatenate your data, but maybe you don't want to concatenate your data because there's different noise levels or different noise processes or different observation models. And you could also concatenate the samples, but maybe you don't want to do that either because there's a batch variable or not. And think about how to represent that is something we've been dealing with for quite a few years, and thinking about what are the right frameworks for moving this forward. I feel one slide there's a more philosophical, what are the different integration questions? I think after the first talk, I can be very brief here. I just really want to make the point that, most of these integration methods we see today, particularly in the sort of single cell view, they make assumptions about presence of anchors. So either we make the assumption that we're measuring the same things, so transcriptome data, in different sets of individuals or experiments. And so the anchor here is that we essentially measure the same entities, the features are shared. And another big integration tool of approaches is where you assume you measure different things, but the samples are shared. So basically where you really make the assumption that for some of the same samples, you're measuring all or subsets or 2 out of 3 or even 3 out of 3 of these modalities. And there's this sort of diagonal integration. I think that's also where Caroline ended her talk, where nothing is shared in common. You neither have the same samples nor the same anchors. And this is obviously the hardest problem. And in practice, many of the integration tasks we have are a mixture of these different areas. And by the way, do please interrupt me at any point in time will be great if you can also at some point get me stopped here and we're just discussing about whatever, however far I got on this presentation. And so I just just to recap, there's sort of different ways of integration. I actually will talk here today really about only this task, which in a way is the easiest in some dimension, because you really assume that you have parallel data, that you really observe explicitly covariation, that you see, observe and measure for the same individuals multiple of these modalities. And this could be also different scales, right? It could be epigenome, transcriptome, it could be genetics, and that's something I will talk about today, but where you really sort of have parallel information, which I think is an idealistic assumption, and perhaps also the easiest, but I think there's also, lots of interesting questions we can solve. So a while ago, we really thought about a class of models that want to reason upon the objects we are profiling. So really, what are the samples and what are the latent spaces? And I think that's also the sort of granularity of the methods we discussed now, where you really would like to sort of align modalities, but aligning them with the sample in mind, understanding where is an individual in the latent space, what does characterize a cell, what is a disease state or a cell state, and it's really about the sample that's sort of front and center. And the one class of models we looked a lot at are linear models, or local linear, actually. So I think really generalizing this sort of trivial idea of PCA that we're all familiar with, you decompose one of these big matrices, which are sort of samples by cells, by features, into smaller matrices. And the key sort of trick was just to have a common latent space, which is essentially, I would say, the local linear version of what you just heard from Caroline in autoencoders. Has some advantages and disadvantages. I think that the advantage is that you get full interpretability for free. The disadvantage is that you have to think about, where to linearize and what are these linear components. So essentially what these model classes do is to think about, in every local linear neighborhood, you're really assuming you can do a linear decomposition and have basically two matrices, one that tells you what are the new coordinate systems that map these factors or new dimensions to your previous dimensions, and the other one, what's the joint representation of these cell samples or features in this new latent space. This sounds like a really trivial basic approach, and I think what's really key about it is its simplicity. But what you're essentially doing is jointly decomposing multiple of these matrices in parallel, where the assumption is that one of the lower dimension embeddings is shared, basically with the new coordinate system or the cells or the samples in these new coordinate systems. There's surprisingly many things you can do with this. You can think about multiple sample groups where we model that not only, I mentioned this already before, we have multiple omics, but also samples are grouped in interesting ways. There's also really interesting questions about thinking about that data now really tensors. We also heard that before, that really what you have today, you have different omics, you have different samples, and maybe even sample is sort of spatially, temporally smooth because you have spatial omics or you have temporal data, and you can really do many of these things. You can really get a lot of mileage out of these types of approaches. What I find is a very interesting discussion point is, where do you want to introduce nonlinearity? Do you want to introduce nonlinearity in the mapping, going from your observed data to your latent space? Or you want to actually do the nonlinearity in the latent components, in the latent space, in your new coordinates? And I think that's a very interesting twist that in practice makes big differences. And I actually, decided to switch gears a little bit and not talk so much about these global integration questions, where the object of interest is the cell, the sample, but actually talk, and that also motivated one of my questions at the end of Caroline's presentation, to really focus on methods that actually don't want to reason upon the samples, but connecting features. And this is a problem, we are having a lot, particularly if you think about genetics. You don't really care about using your genome data and your phenotype data to define a latent risk state of an individual, which you can, but mostly, what we're doing today is actually trying to find the relationships between these different features. For example, correlating genotype to phenotype. We have a property of an individual, which might be its makeup at one position in our millions of common variants we can assay now in whole genome sequencing. And we want to correlate this one property with another property, which might be his or her phenotypic state in one dimension, which might be as benign as expression of a gene, or might be as severe as consequences of risk for diseases. So it's more think about how features relate to one another. In that way, I have here sort of two, examples of problems we are thinking about a lot right now, and that I think are also interesting to consider for these, joined up efforts that Melina was really nicely introducing. The first is thinking about, how do we deal with rare variants? And I just have a few introductory slides for those who don't think about rare variants. day to day. We're all sort of fascinated by this concept of GWAS. We can essentially solve linear regression, which is still, I think, what we mostly do today. And the idea is we're correlating every single variant in the genome with phenotypic outcome. And if you do find an association like the one here depicted at the top, you basically have a linear relationship between a particular makeup of a genetics and a trait. And if you look at GWAS today, like, for example, here on the right, you see the effect size. So basically, what are the regression coefficients you're fetting as a function of allele frequency? This is for height, and it's very common that most of the action, if you think about your own personalized risk, is for these variants that are extremely rare. So rare variants are not as strongly under selection, or rather, they've not been weeded out in the population. That's also why rare variants do have large effects. And there's really this interesting question: how do we get at these rare variants? That drive much of the signal, that drive much of actually your personal risk for disease, and where also effect sizes are large enough that you can actually do any inferences, because many of these small effects here are very, very difficult to deal with. Think about joint embeddings. These effect sizes are very attractive for a number of reasons, and you really want to get a handle of them. Now, the problem is, rare variants is a difficult task. These variants are sparse. There's few by definition. The genome is a big place. For common variants, for rare variant becomes even larger. And what the sort of solutions are sort of twofold. The first is really we do typically region-based testing. So we take not variants one at a time, but rather, for example, look at the load of carrying a particular number of impactful variants in a region of interest. And for the sake of simplicity, I want to assume here the region of interest is a gene. And so essentially we're just summing them up. This guy here, for example, has one region of interest, let's say in the coding region of a given gene. This guy has another impactful variant, but it's a different one. And this individual here has two impactful variants that are here indicated with red. Now, the problem with this is you need to know what are impactful variants. And what we do today is typically use annotations, we use rare variants, we utilize annotations like missense annotations or predicted splice features. And I come to that later, but the real interesting question is how to actually use these annotations in a flexible way to understand what are impactful questions. There's another class of methods that I don't want to go much into detail. Variance component tests. Many of you may sort of use SCAT and other approaches, but essentially these are still linear regression models where you assume sharing of effect size, or you marginalize over them to be precise. That mitigates some of the sparsity, but at the end of the day, there's similar issues. We think about how to annotate which variants, integrate them, and use which annotations to weigh them and to utilize it in different strength. And this whole field lives off the ability to integrate and handle annotations appropriately. Because if you just sum up a number of variants, you get nothing. There's no meaningful burden. It's really important to implicitly what this vector is meant to represent is the gene disruption score. How much perturbed is the function of a particular gene? Maybe you think about how much it approaches a natural knockout, which we do have, right? In particular, in loss of function variants, which do exist, although at a low frequency. So these are the limitations. I think I already mentioned them, pretty much all of them. Annotations are difficult to get. You have to think about how to burden them, how to filter them. And what we wanted to do is to think about, can we sub-release it with a data-driven approach that makes very, very few assumptions, that basically just learns the rules about which variants with which annotations are impactful. There's a few subheadings that we were also interested in. One is nonlinear effects. And maybe also think about phenotype predictions, but I really want to draw the attention to this task, this quest of using annotations in a flexible way without pre-describing the solution to the problem you're trying to solve. So I mentioned already annotations, and I think I should sort of briefly introduce where these annotations come from. So we're utilizing everything that today's genomics, genetics community is producing. There is exciting progress and methods for predicting splicing. For example, there is traditional approaches like variant effect prediction or deep C, where we now can look at regulatory effects of virtually any encode track. What is here not on the slide, which we're actually now incorporating as we speak, is things like alpha missense, which are computational predictions for missense predictions. But what these things are, and we have now a real compendia of models, essentially models where you can ask, well, give me a variant, and I want to know what's the impact of a particular change from the wild type or reference sequence And these models are going directly from sequence to some readout, which might be missenseness or splicing or whatever. And if there's a large predictive effect, then this variant is likely to be driving that particular molecular layer, driving a splicing differences, being associated with loss of function, or affecting gene expression, if you think something like an informer model. So you can use whatever you want. And what you get at the end of the day is a number for every mutation. And that number is some measure of the extent to which a particular model believes a particular variant is associated with a particular function. And now, once we have set up the data, what we sort of set out to do, and this is really work from Brian, a postdoc in the lab, and Eva Holtkamp, who did her master's project with collaboration with Julien Gagneux. And there's also a preprint at the bottom that you can take a look at. And what we sort of set out is asking, well, if we have variants and notations, can we not just treat this gene impairment as a latent variable we have not observed, we don't know about? We would like to really go from annotations of variant to what is that? How much is a particular gene and particular individual impaired due to their individual genomic profile? And once you have those impairment scores, then you can basically predict phenotype. And we don't do this only for one phenotype, but we train this model actually for multiple phenotypes at a time. So essentially it's going from variant annotations to trait, to gene impairment, to trait prediction across a set of traits. And I show you later what the specific traits are we're using. Now there's a number of considerations, but the key consideration is this gray box here, which basically tells you how do you go from variant annotations to gene impairment. And here we're using a concept that has different names. Deep set is perhaps closest to the annotation we're using, which is a New York paper a few years ago. Multiple instance learning is another name of the same concept. Essentially, we're learning aggregation function that says, well, the individual personalized variant profile of an individual particular gene is aggregated using a neural network that can basically really learn the rules. Learn the rules of, we threshold the values at a particular annotation levels, we summing them up or we averaging them and so forth, and to derive really at this sort of overall gene impairment score. And intuitively, as you can think about it, if the score is 1, it means the gene is heavily impaired, and if it's 0, it's not impaired. In honesty, we haven't really worked on the calibration of these scores. So what these scores at the moment do, they're relatively calibrated. They capture the inter-individual variation impairment, but perhaps they're not designed to capture absolute impairment at this point. But as we see later, that really doesn't matter, because what you care about is just understanding how much more is one gene in one individual impaired than another gene, than the same gene in another individual. That's what you need to look at association and differences. So that's the approach we're using, and this can basically tag in any annotations you have, any variant profiles. And variant allele frequency, by the way, is just a notation for us. So the frequency of a variant in the population is annotation like any other annotation. Now, there's a twist that I want to just mention. This is at the moment, the way we train this model is we actually introduce a set of genes for which we already know that they're associated with a trait of interest. And this has a one really nice example, namely that you can assess holdout genes that you're not using for training without even worrying that you're double dipping. So the testing results I'm going to show you, they're always evaluated on genes this model has not seen during training. And the model can obviously generalize to new holdout samples that you have not seen during training. But the key point, even the genes, we don't see. So we train this on genes that we know are associated, for example, using existing rare variant association tests, and then train this. And then basically, once you have that, you can hold the model fixed and then solve questions of interest. For example, one question you might want to ask is, is a particular gene associated with phenotype? So basically, you can evaluate this model now, new gene, and ask, how impaired is this gene in individual 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so forth? And you get a continuous vector of scores, and you essentially can associate phenotype. And this is as difficult as linear regression. It's the simplest rare variant association test imaginable, because for fixed parameters, this model is lightning fast to evaluate and basically predicts the impairment of a particular gene. And you can use this for phenotype association testing. But you can also use it for predicting phenotypes, right? You can easily train a predictor. You have whatever 20,000 protein coding genes. So you have 20,000 of these vectors, and you can directly use them to predict phenotype. And I think there's hope to assume that such models are useful for particularly looking at the extreme end profile, the rare variant profile, where large effects play a matter and where maybe polygenic risk scores come to their limits. So here's a few results to sort of show you how well this works in practice. This is trained on 21 traits in UK Biobank. So these are mainly blood traits, but also a number of other traits, including height and a few polygenic traits. We use 170,000 individuals on the whole exome release. And the reason we use 170,000 is that you can actually assess these models in terms of the replication on the remainder of UK Biobank. And what you see here is basically for a number of traits, how many associations do you find? So these are individual gene trait associations. And what I'm comparing to, I think what's most relevant is what we use today on every day, which is something like a burden and SCAP method, where you combine this particular missense and peel-off thresholds. There are a number, or actually two notable approaches. One is STAR, and the other one is method by Multimodal, that also use annotations. But these methods are actually meta methods. They're combining different thresholds, different annotations to do those. And I think what I want to convince you with this slide is that this one model, which essentially is linear regression of a single gene burden that is trait agnostic, is as good as complicated meta models that are extremely not insufficient in their complexity computationally and generalizability, which I can show you later, to train. So you find as many or more associations. And what I show you on the right is actually how many of these associations do we replicate in the remainder of the UK Biobank cohort. And what is shown here, you rank these associations across all traits by significance. And then you're asking, to what extent can you replicate these associations in the full release of UK Biobank cohort? Actually, this is using another standard linear association model. So not using our model, but essentially a conventional peel-off burden taste approaches. And you can see you replicate at different extents, but these methods that use annotations do quite a bit better in this regard. So I made this claim that this burden is just one number and you can use it wherever you want. And one way to test it is actually apply the model to other traits, and you can do this as well. So here, basically, if we took this model and just use it per classification for other traits, so 13 additional traits that were not used during training, and you can see these are sort of another disease traits, diabetes, liver diseases, physical measures, renal, and so forth, and you do get quite a bit bump in terms of discovery compared to other approaches. Now, what I find is actually the interesting dimension is that this is getting you really a handle and making predictions for traits. What we've done here is basically said, well, if we have burden scores for just genes and individuals, you can just take those and predict traits quantitatively and ask how well do you do. And what you see here is the improvement compared to a polygenic risk score. Polygenic risk scores that are pre-trained by others that only lose common variants. And what you see here is there is a relative R-squared improvement in percent. So what I think is important to take away is, once these numbers are tiny, and that's not unexpected, because if you think about my first slide where I showed you, you have large effects, but in few individuals, and because they're in few individuals, the variance explained component is small. And I think there's an intrinsic property of many of these rare variant genetic approaches. So it's a small effect. But I think what's also very interesting to see, if you train a burden, based on SIFTs, place AI, peel-off, and so forth, you get a significantly smaller bump than if you use these burden scores from deep RBAT. You can also train other questions where you want to predict the top or bottom 1% individuals. You get a similar picture that you can look at, particularly for the top individuals. You can see an appreciated bump in how well you can predict these individuals at high risk. So this is maybe not the most straightforward visualization. I think this is a much nicer way to look at this. What you're basically showing here is, if I take all individuals and I rank them by the predicted difference compared to PRS, and I ask, do they have extreme phenotypes? Yeah, you can really see this enrichment for individuals with extreme phenotypes is really quite nicely seen. So you are able to really pinpoint individuals with extreme phenotypes based on the predictive signatures. You can train using such a model that leverages the rare variant profile for individual genes and goes about those. So this is, I think, quite a universal tool where we can think about how we use rare variants in a way, and I think it's just one dimension of integrating annotations that I think would be quite also interesting for efforts and studies like the one that's maybe interested in this community. Maybe I pause here quickly if there are questions.
I do have one. I just wanted to see if I understood correctly. So this that you presented, are we talking about variants in genes or coding regions, right?
Yes. So it's absolutely correct. So at the moment we're looking really at variants that are coding or near coding. So core gene promoter and splicing variants are included. Reasons is simply the complexity of mapping enhancers to genes and non-coding space is something we have not considered. I think it's a very good follow-up question that at the moment we're not considering. It's a very good point.
Okay, no, I mean it's very great because this can be used. I can see how you can use this by potentially predict if a certain new patient might have, or what is the possibility of getting a disease. This is incredible. So do you think you could perhaps use just something simple, like say taking plus or minus or whatever 2 kb around the gene and include those variants as belonging to the gene? Could it be as simple as that, or could you?
Yeah, I think if you really. Yeah, I think if you're really near the gene promoter, it's fine. But as long as you go to distal regions, and you need a good mapping of enhancers to genes. And the other dimension is, I think they're no longer a universal, score column code works, because you need to really think about variants and what is a positional relationship to the gene. Yeah, and that's the aspect we don't have to care about this here. So I think it needs some serious thinking about if you really think about the non-coding space, but near coding is very doable, as you're alluding to, yeah.
And I do have another one. If nobody else, I don't want to take, but I do have another question.
Just go, and you should go ahead. Yeah.
Okay, thank you. I'm wondering if with this tool, you're able to interpret your variable and actually go back and do some kind of 5 mapping. Because right now you're taking everything together, right? You're looking at the common effect of everything. Is it possible there is a chance you can look at the variables, the parameters, and go back and try to do fine mapping and call the rare variants, or it's something that cannot be done with this?
Yeah, that's a good point. We haven't done that, to be honest. I mean, we looked at what's the function, that we're learning. What does this function look like? And obviously, it prioritizes the things you would expect. It loves its rare variants, and it loves peel of annotations and so forth. But you could also play the other way around and basically, look at the relevance of the variants for a single gene for an individual. That's certainly totally possible to re-annotate the relevance scores, which is something we haven't done. Yeah, it's a very good point. It's straightforward.
Thank you.
Any other questions on this part of Ollie's talk?
So if you go back to the previous figure, Ollie, I guess, sorry, this one, yeah. So the relative R squared, so this is like the improvement in prediction R squared?
Yes, compared to PRS, yeah.
Okay. Yeah. Yeah, it's
I mean, I think it's important to be honest about, that obviously improvements if you measure them by R squared will be small, right? Because these effects are rare and, but they have large phenotypic effects for those individuals that carry these variants. And so I think it's a question of which metric you use. And I think that's just the wrong metric, but I think it's important to also be mindful about this.
I'm wondering also, like, when you fit these polygenic models, could you fit these simultaneously as some yeah
totally imaginable. I mean, this model is in a way self-regularizing, because what we have to think about, and that's actually also quite different to other rare variant tests. If other rare variant tests, you also worry whether they're contaminated by common signals. We don't really have this issue, at least in practice not, because we assume that it's a universal set of rules that predict impairment for every gene, for all genes across the genome and across all the traits. And so, it's not gonna, but just tag a common variant signal in the same way than a normal burden score does. It doesn't have this issue. At least if it is not explained by the sort of rules that are captured by this function that is learned. However, I think learning it jointly makes sense. It's totally on our list to do this. If you think about it, there's these two worlds. You're either trying to learn a universal rule from variance to gene disruption, or you're really memorizing what's the variant profile. Let's say a really common GWAS signal was one variant, present, absent, yes, and you're training linear regression to a phenotype of interest. And I think the benefits of these two approaches, they cross over as a function of the frequency of the variant. If it's too rare, you can't train your linear regression anymore. And conversely, how accurate these annotations are. The better we have models that are able to predict variant impact, the better these models will also generalize and also tap into the common variant space. Right now, these annotations are frankly still too poor to really be competitive with conventional regression methods for common variants. They're only competitive when you can no longer train regression, which is the real variant space. But I think this will cross over in the future, and I think these worlds will meet, which I think is what you were implying by your comment.
Yeah.
Okay. Well, I think I will just
maybe last question. Sorry.
Yeah, go for it. Go for it. No, we can discuss it as long as you.
Yeah, sorry. Do you think it would? So say you have so there's like these methods to impute the summer statistics, right? Could you impute these in summer statistics? Maybe I just haven't thought about this. It's like coming from.
Good question.
Right.
It's a very good question. I mean, I think if you're moving towards a space where you have summary statistics of sufficiently rare variants, you might be able to train these models. It's a very good question. That you could train them directly from summary statistics. I mean, that would be quite interesting, training from summary statistics and then going back into a cohort and using this pre-trained model for association testing. We haven't tried that, but I think it will only work if you really get, summary statistics that basically max out the allele frequency distribution and go into the extreme rare cases. I would expect the moment it's not going to be competitive, but this is a very good question. Yeah. Thank you.
Thanks. Sorry.
No, that's great. Yeah, so the second piece I sort of wanted to share today is, we've done obviously a lot of work, once you've found this association, common or rare, one of the question is what are the functions of these variants? And I think this variant to, function mapping, and that we heard also in the introduction is sort of key interest. So for now, I will assume we have these maps, we know what are the variants, and the question is, what do these things do? And these are things we have to really discover in large cohorts like UK Biobank or other cohorts of this size. Now, when we go to function, the good news is it becomes a lot easier because linking variants to molecular outputs is actually something, and we have been demonstrating in the field that this is viable, from cohorts of maybe 100 people. For example, if you look at expression quantitative trait loci, we now correlate the same variants to a difference in gene expression in a particular tissue. So it's an easier problem, and the reason is that obviously expression is just a much simpler trait than a disease endpoint that might manifest somewhere late in life. However, the challenge is that this error here depends heavily on cell type, cell states. So variants may exert a functional effect on particular cell types, on gene and particular cell types and cell states. If you really think about what is the right cell type and context to measure these variant effect in. And so here's a different diagram of this problem to represent that. We want to go from variants to maybe gene effects in cis, and this is actually what I will really focus on here in this talk. Probably for time interest, I'm not going to go much further. Where we really want to map what are the relationships between variant in the known coding space, particularly to the next regulated genes, and then there might be downstream consequences on pathways and cell types downstream. How they modulate and pathways that are regulated and then ultimately propagated to disease phenotypes. And these paths and these graphs will be vastly different across cell types. So this error may just not be present. So we might not be able to detect a particular variant effect in one cell type, or it might be linking to another gene, or the downstream consequences might be different. And this sort of motivates the use of single cell assays, which is something we've been quite excited about. So this is a picture, apologies, I borrow from the lifetime perspective article from Nicola Zarevsky. I think it really makes nicely this point about that if we think about, the sort of cellular states we're capturing across life, it's both developmental axis where particular progenitor cells then manifest in differentiated cell types, but it's also different trajectories in disease progression when cells take the wrong turn and disease emerges. And single cell readouts obviously fantastically place to sort of capture many of these transitions in a continuous manner without having to worry in a very, very fine-grained detail, where do you sample, when do you sample, and what time in life do you sample. And so we have been, a few years ago, started quite a program to try and create the data in this area with collaborators. Here's just one example that I want to use. Now there's much larger data sets, but this is in 215 human iPSCs from the UK Human-Induced Pluripotent Stem Cell Initiative, where we had basically healthy reference normal individuals, and we subjected them to a differentiation time course with neurons and collected using single cell readouts cells at day 11, day 30, day 52. There's lots of tricks that I want to glance over here today. One of them is pooling. So actually this whole study is only 10 experiments, and we basically pooled individuals of diverse genetic backgrounds to improve efficiency. But what it really boils down to is that at each of these different stages of differentiation, we can now look at, what are the subpopulations, and what we can now look into these UMaps that we're all familiar with in each of these cells. Here in the beginning, these are blue populations, progenitors, that then later on turn into these green guys, which are neuronal origin of different types. But at each of these different subpopulations, we know from which of these 250 individuals does a cell come from. And that allows you to start interlinking the relationship between the germline genome and cellular programs and cell types and gene expression in these different cell types. And this is essentially opening up the possibility to do association genetics. So we can take, here, for example, for the example of dopamine neuron and serotonin neurons, we can take the same individuals, pool cells by donor, and then ask whether those individuals that carry a particular allele are associated with increased or decreased expression. Here's one example of a gene PPA2C, and it doesn't really matter what it is, but this is a coding variant of this gene in the promoter, actually. And we are asking now, how does different allelic states, so every dot here is basically a person, and we're collapsing all the cells from a particular donor of origin, and how does the genetics basically correlate with the expression phenotype? In this case, there is an association, and there's no association in these other cell types. So we can do everything we want to do, right? And so map cell types and map QTLs, one by one. We can do this here for these two cell types. We can do it for all the cell types. We can call QTLs as if we had conducted an expression study in each of the individual cellular substate. In this case, that's the stage of differentiation and its cell type and map QTLs, how we have them. And if you care about, you can also think about nice co-localization examples. Here, for example, the hit where you have an association in day 52, that's correlated with a gene of schizophrenia, and you can look at co-localization events. But I don't really want to talk about this here, but really ask, how in the future we would like to analyze these studies, because obviously, if you think about this UMAP plot I've shown you, this is deeply unsatisfying, right? To sort of call fixed subset of populations by annotation and collapse them. Would really like to utilize this whole manifold of cell states in an unbiased way, to think about where does genetic regulation act and how do they co-vary and modulate expression changes. And that's something we've been doing for a number of years. This is a work from Anna and Tobias that we're also still extending in different directions. And the whole concept is that we do not want to having to discretize your cell states. What the approach I just presented to you is basically we take cells, give them annotation, for example, in the differentiation context, or I've shown you one where we look at different neuronal subsets, and each of the cell in this annotation context are collapsed, and then we ask, is there an expression association between particular variants and gene expression in this population, this population, or this population? And you can find, for example, no association in these low guys, and an association that is stronger in the middle differentiation here, and even stronger association in these highly differentiated cellular subsets. For example, let's imagine these are iPS cells and these are neurons at different stages of maturation that we want to like map. But what we would really like to get to is a setup where we don't have to worry about the annotation. And this is here in this cartoon illustrated as we want to estimate genetic effects in every cell. Here, basically, again, cells are colored by the effect size estimates, but regardless of their annotations. And this is obviously an undefined problem because you can't fit a regression model to the counterfactual cell that you haven't observed. Each cell is associated exactly with one individual and with one genotype. So you can't ask what is the genetic effect of a particular variant in a given cell, simply because you only have one genotype in this specific cell available. But what you can do, and this turns out to be very fruitful, is you can essentially use a totally over-parameterized model to solve this problem, and leveraging the fact that these cells are not independent, but they covary in terms of their regulatory programs, in terms of where are they similar transcriptionally, where are they dissimilar. And so what we do here in just one piece of an equation is essentially we fit a normal expression model, so where y is now this sort of long vector of expression levels of a particular gene, say. Every cell is one entry, so it's a cell long vector, which you see sort of here on the right. And then we fit a persistent effect that says, this is just the effect of the genotype of that cell, independent of cell state. That just says, there's a shared component, and there's an effect that is invariant to cell states, maybe to use also some of the causal representation that's actually also underlying these ideas here. So this is the invariant component, if you wish. But then we have effects that are specific for every cellular context. And those we fit by saying, well, now every cell has their own effect size vector. And this model is not making much sense because you have as many parameters as you have data points. You have basically an effect size vector that is specific for every cell. And so you have cells numbers of parameters and cells numbers of observations. So naively, you can't really fit that. But the idea is now you can regularize this guy here using a cell state covariance matrix. Essentially, we're regularizing a multivariate normal distribution, that basically encodes the idea that cells that are similar in a cell state space, they need to covary in the effect state space. And that's something I just want to quickly illustrate at the bottom. For example, here you would have a covariance matrix that captures discrete cell states. So group 1, group 2, group 3. So this is the low, medium, high. But you can also just dump in all the information we have from cell states today. For example, a representation from an autoencoder or just PCA, and essentially measure or estimate the total covariance in cell state between cells. And that essentially changes the defective numbers of degrees of freedom in this linear model. And this is because it becomes identifiable. So that's an embarrassingly simple trick that does work quite well in practice. So I want to show you here just an example. So we only look at neurons. So I only look at one blob of dopamine neurons. So basically it's this green population here at the top, which previously we just considered this one cell type. And now the key question is obviously whether there is sub-state specific regulation, whether genetic variants exert different effects depending on where in this blob a particular cell resides, quote unquote. And this is what you get from applying this approach. So here on the left, you basically see the sort of a T-SNE representation of the cell states. So these are all the dopamine neurons, at day 30, at day 52, and these are those treated with a stimulus condition. So you can see here the cell state representation. On the right, what you see is the estimated effect size of one particular variant, an EQTL, on one gene. Again, I use this purely for illustration. But the key point here, this is not the expression of this gene, but it's the estimated allelic effect. And you can see it's only estimated to be active here in this bottom right population. What we see is roughly a quarter of all genes of all EQTL have this sort of cellular substate specific effect. So a quarter of all EQTLs with evidence to be active in dopamine neurons are substate regulated and hence are exhibiting genetic effects that are only visible in some of these cellular cell states. Now, what you can do next, you can ask, well, how does it look in general? So what we did, we clustered those effects, and you can now really observe that there's roughly 10 different prototypes of where gene regulation occurs, either here in this DOP population cluster here in the bottom or elsewhere. So there's really sort of manifold different regulatory profiles that even within a cell type, again, just to emphasize, the within cell type regulatory effects that manifest in sort of fine-grained differences that we can start now to appreciate and to dive into. And what I want to just make a note, maybe on a philosophical comment, one could even argue that this is the way we would like to identify cell types. Basically not identify cell types by annotations or other means, but essentially, if there is a population of cells that recurrently across different common variants is regulated in a specific way, that also may help us actually to redefine what cell types are. So this, you can really turn this whole problem of cell identity definition upside down and first look at where does regulation occur. And based on the inference of where regulation occur, then get to maybe also improved cell type annotations. I just want to make one comment, that many times there's a question, well, you could just, subdivide the space more fine-grainedly. And, I showed you these 3 sort of cell states that are purely caused by the experimental condition. Obviously, you can also cluster the cell space into 9 groups or 18 groups. And one obvious question is, to really benefit from having this continuous representation. And here on the right, you see always a scatter plot between this continuous scale representation on the x-axis. This is the significant organizations we identify versus discrete. And, you can see there's always a significant bump. Actually, at some point, it saturates when you have 18 cluster as well, these continuous representations that leverage the co-variation across cell types do significantly better. And I think you can start to appreciate in this setting here why that's the case. If you ever more fine-grainedly subdivide what a cell type is, you're losing out in sharing statistical evidence strength across related cell states. And this is one of the reasons why, fine-grained discretization is inferior to finding such linkages and correlations between cell states, while still accounting for the fact that there's differential regulation between these different subgroups. I don't want to go here in details, but what we also find is the first evidence that actually these more fine-grained, fine-mapped substates also useful for co-localization. I think this is quite interesting. If you think about it, we're really interested or used to this question of fine mapping. We want to map a GWAS hit to a molecular output, and essentially what we now can really start to be doing is fine mapping in 2 dimensions. We would like to fine map what are the causal variants, but would also like to fine map where is the cell state and the cellular subsets where these associations occur. And I think that's a very, important area also for the future, to leverage the resolution of these data to really sort of do 2-dimensional fine mapping, if you wish, in cell state space and in genetic space. And what I want to just show you here is that if you get the cell state space wrong, you activate a lot less power to do the genetics and vice versa. So there's a close coupling between doing the genetics and understanding in which substates these things to reside. Yeah, and this is essentially the main bit I want to talk to you today. I think I showed you this deep RBAT integration approach for leveraging functional annotations, and I think it's a nice tool for bridging in the whole world of GWAS, predictive models, methods that are now with increasing accuracy able to predict functional effects of variants on splicing, gene expression as a whole, missenseness or otherwise, but still leveraging them in a GWAS setting, and in particularly also overcoming some of the circularity that's often associated with end-to-end deep learning methods. By just splitting data appropriately, you can basically get the best of both worlds. You're still conducting association genetics, but you're losing these predictions in a flexible way. And the second part, what I talked about, is population scale single-cell sequencing and how we can use this for really going from variant to function, and what are methodological challenges associated to this. I talked about these real questions about how do we go from variant to function, and what I just want to really emphasize, I really focus on this first bit of this arrow, from variant to the first gene. There's a lot more to be done, to go into future understanding, what are the pathways that are associated with this, and I think that's really where we need perturbations, and this is future for us, and I think that's also future for your thinking, where we are now with here's an example from the US Parse Lab, where we're now combining actually population scale variation, where we have cells, iPS cells from different donors, and then we perturb them using CRISPR. And this will really allow us to go even further and understanding what are the downstream consequences. And we are really fascinated right now also of thinking about causal inference methods. So I think I'll skip that in the interest of time, but very happy to also discuss if there are questions. So what we are really thinking about is, going basically on the one hand to total causal effects. So for every cell, we know which individual does it come from, which perturbation does it carry, and also look at which genes are affected by a particular knockout, but also going to really directed causal networks where we can really understand, what is the causal order of events, variant to gene to pathway, And I think that would be very, very interesting for them taking, you know, this more holistic views and decoding networks. But maybe I stop here and I take questions. Just before going there, I just would really like to acknowledge. I skip that. I really acknowledge the lab. There's many people involved. I think I acknowledge the people as along through the slides, but I also want to highlight a number of collaboration with Dan Gaffney's lab, with we did the IPS differentiation, Leopard's lab, and also Nicole Soronso, with whom we're working on scaling this right now to actually UK Biobank scale single cell sequencing, which I hope I can talk to you about next. So I think I'll stop here and we can discussions, and maybe if people are interested in these sort of causal inference methods, I can show some more slides in the context then. Thank you.
Thank you so much, Ali. I just wanted to thank you again, and also say that it's so exciting the directions the field is headed. Those last few slides that you outlined about you know, the population scale data and the perturbations on those data. I think that's just where it's clearly where the field is headed. It's very resonant with the goals and directions here on our side of the ocean as well, and in the center. I guess the center's on both sides of the ocean. But yeah, so thank you. I guess we should open it up for questions. Bjarni, did you want to say anything as well?
No, I wasn't. I don't have a question if you're asking that, but
okay.
But I can ask something.
No. I think I mean, anybody can open. We can open it up for questions. Genio, did you? I see you.
Yeah, I mean, I do. It was very, very cool. I really, really liked the talk. I was wondering, and I mean, I work a lot with shared-seq data, which is, you know, RNA and attack. So that's why I always ask you about the attack signal around. So I'm gonna ask again for this last thing you mentioned about finding these subclusters, whatever we want to call it, I don't know, with genes that are more active in just a subset of that cell cluster. Do you have multimodal data? And the idea is that, for example, if you find at day 0, I don't know which day you had, I don't remember, but day 0, you have actually a transcription factor coding gene that is, you know, modulated by a genotype. Then you can look at the attack accessibility, and technically you should see that the environment, like the accessibility around the genes that contain that motif for that transcription factor, should be changed if that is really affecting the trajectory, right? Because you have, so basically the transcription factor that changes expression needs to bind somewhere in the genome. So if you have paired attack, you should see that in the same UMAP. If you plot the accessibility for those sites, you should see something similar to validate. Does that make sense? Or would it be possible?
Yeah, no, it's a very good point. I mean, there's now data coming where we have paired attack, actually multi-ohm data from differentiating iPS cells with brain organoids, where we could look at that. At that time, we don't have the data, so we couldn't take these inferences. And what we have done is looked at bulk data set in another context of different stages of differentiation, where we could exactly see what you're describing. You know, that basically the attack profile foreshadows also the expression QTLs, and you can really look at the order of events, if you wish, particularly of the cell-specific effects, which are what we really care about. And I agree that the attack will be a phenomenal asset to do these things right. What we are actually now using is methods for tying together attack and RNA, to define a cell state in the first place, because what these methods all rely on is having a good cell state map, right? And I think it really helps if your cell states capture or reflect biologically meaningful interpretations. And this is, I think, a bit of a chicken egg problem. Do you first do inference and then annotate what they are? Or do you want to, have cell state representations that capture regulation? And particularly for the multi-ohm RNA attack, this is, I think, a fantastic opportunity to basically prime these models towards capturing relevant cell states, activity of transcription factors, and other dimensions, which is doable because we have this regulatory component baked into the assay. So I think that's a very good point. And this is a starting point where we think we have to think harder about how to make that work.
Yeah, we have the same problems of the integration and attack. Actually, I want to correct what I said before, because actually we see sometimes that we see a change in accessibility before we see a change in genes. So potentially you should see the attack changes for the gene that is activated later. You should see it before in the previous cluster. But those are, yeah, that we see the accessibility changing before we see the genome changing, which kind of makes sense, right? It takes a little bit for all this cascade to take effect and actually see the gene changes. But anyway, no, that's very cool. Thank you.
Ollie, you mentioned that you had a couple of thoughts or slides about the causal inference piece, but it's entirely up to you. If you'd rather wrap up, that's very understandable.
Yeah, no, I can show you some bits which I glanced over. So, what we're at the moment doing, is really think about how do we get directed causal networks? I sort of alluded to these ideas, which that we want from variants to genes, but then really understand what's going downstream, and the questions obviously how to represent this, what are the right tools, and it's a bit of a different theme because we don't want to learn a causal representation, but really a graph that captures gene regulation, which is I think a bit of a different task than what we heard earlier this afternoon or morning. And so we have been really thinking about, what are the right model classes. We tried a long time with, acyclic models that we are all thinking about, fitting directed acyclic graphs, which is, I think, very popular. And I think it has been a very good starting point. In our hands, this has really failed with the exception for a couple of extremely linear pathways. So we had these few examples where, hypoxia and others, where you have a very, very linear pathway. There's a root node and everything happens downstream. But in general biology, is unfortunately, even in practice, not only in the textbooks, but also in the inference, extremely circular and cyclic, where you have genes regulating others, doing self cycles. And that makes it obviously really hard to draw these inferences. And the way we have been using this right now, and there's been a lot of work also by others in the field to think about unraveling this dynamics and basically understanding what is the regulation from a particular time point to a derived time point, and thereby basically having a directed graph across the sort of linearized time point that we can then describe using a structured equation model. And this is, I think, an interesting complexity intermediate. The extreme one is to really look at the full dynamics, combining velocity ideas with regulation, which is a hard problem. But this here seems to be tractable. So we have now some work that's submitted where we are basically using the structured equation model, but have additional information that tells you which is the perturbation. Yeah, if you basically perturb a particular cell, a particular gene, a particular cell, you basically the shared equation that's actually really embracing this invariant mechanism principle that also Caroline was mentioned earlier, where we assume we have the same dynamics that explains all the cells in the same way that is shared across your whole population, except for these perturbations that we know that, for example, one cell has a single gene perturbation or a double gene perturbation where this dynamic system is perturbed and not shared, because if you knock out a gene, then that gene and all the parent genes, its expression will be affected, and your violation from the sharing. And this works now. Actually, it's the first time we have something that at all works in the space. We can now recover similar graphs, which I think you may care about or not care about. But we can, do quite a bit better than other methods, including actually methods that use neural network-based approaches or make less assumptions, but in our hands don't work so well for these types of questions. And they can also make predictions of new holdout perturbations. So we can really train this model and ask, let's give us a few perturbations. We train this model that knows which cells are perturbed, which genes, single or double perturbations, and then we can really understand, what's the predicted effect of an unseen knockout perturbation, a gene knockout perturbation. And this is a metric we adapted from the community. I think this work from Rohanidal and many others have now using this and asking the right metric is to basically look at what are the top differentially expressed genes if I knock out a gene. I don't care about the change in distribution. I care about the really strong effects. And there we at least see in our preliminary results as early days, we see quite a big boost in accuracy, that we are able to reduce the error quite significantly using these types of approaches. So this is at the moment where we are with that, and I think that will be really the sort of the stepping stone, to tie this all together with precision genetics, and then go end to end and look at variant to function, but then also understanding what are the downstream networks and how they're perturbed, which is definitely work in progress. But I think that's what we're thinking of right now.
You hinted at an idea that I thought was very interesting, which was that somehow instead of the usual way of annotating cell types by where they sit in expression space, to think about it as actually using the regulatory structures to define that, and that you hinted at that there might be a way to do that continuously rather than discretely. I thought I heard you saying that sort of thing, and if so, could you say about it?
Yeah, no, I think in this one example I showed, where we are, looking into mapping the effects of common variants in a continuous manner across cell states, not defined by discrete notations. And I made this comment that I think it's interesting to think about what is a cell type, and whether a cell type is not defined by the response of cells to a perturbation of whatever type. And in this case, we look at natural genetic variants, which are variants we carry, common variants in the population. One could and maybe should, even consider definitely perturbations in this context of chemical or genetic variant. But in a way, I think it's not only a matter of modeling the perturbation effect independent of cell type annotations, but conversely, we should maybe define cell types based on how perturbations pan out, because this is a much closer to function readout. How cells respond to how
exactly.
Then their position.
Than just clustering them in the state space. It's the functional definition. It's not just where are they, but what do they do, and in response to what that should actually define what they are, right? I mean,
Yes.
No, I think okay. So I
think it's. Yeah. If we have enough perturbation information, that's a question in a way, how much variation we have available. But the richer perturbation data we have, the more viable that will be, to basically cluster cells by their response behaviors, rather than by the pure transcriptome space. And I think that will actually be quite enlightening in some ways. Yeah. We definitely see already that, that cells that are similar in the transcriptome space don't, or clothed by some definition, don't necessarily have to be super close in the response phenotype. So I think it may change our understanding quite significantly.
Right. Yeah. It's not just the categoricals. It's the sort of the metric, the spatial, the using the responsive perturbations to actually define the space of types in a more meaningful way, biologically meaningful way, I guess. That I thought you were saying something like that. So I think that's a very cool idea. Seems like the right direction. Any more questions? We should definitely let folks in Europe, including you, Ollie, have their evenings. But thank you so much. Thanks so much for your time and for your presentation again.
Yep, thank you for having me.
This was really, really cool. And yeah, I guess we look forward to seeing people tomorrow. And who's starting again tomorrow? It is, let me see.
We have Jesper. Oh, it's Jan. Yeah, I know it's Jan first, and then Jesper, and then we'll have Adit doing the hands-on session. Anyway, we'll look forward to seeing many of you tomorrow.